Hi, it's Moser with a quick tutorial on the ecology of populations. So, after you've watched this video, you should be able to identify 3 things biologists measure about populations. You should be able to distinguish between dispersion patterns, and you should be able to explain how density and dispersion patterns can affect population sampling. You better already know what a population is, but just in case, we'll review. So what's a population? Well, remember, it's all the individuals of the same species who are capable of reproducing in a given area. So we could talk about the red oaks in Beaver Creek State Park as a population of red oaks. We could talk about the common snapping turtles in Pymatuning Lake. Calidra serpentina, to get all, genus and specific about it. We could talk about all the possums in southern Columbiana County. Didelphus virginiana. Aw, isn't it cute? Not especially. Those are populations. And in this chapter, we're going to focus on populations. Biologists very often focus on individual populations in a larger biological community to see how they're changing. So, the material that we're going to start doing is mostly out of Chapter 20 in your Holt Biology textbook, and it's technically called the Ecology of Populations. So the first thing we have to do, which we just did, is remind you what a population is. Organisms in the same species living in the particular same place at the particular same time. But you knew that. So there are 3 different things that there are actually probably hundreds of things that biologists look at about populations, but we're going to start off talking about 3 of them: population size, density, and dispersion. Because looking at those 3 factors gives us a window into how populations change. So, population size is the thing that everybody thinks about when they talk about a population, and that's just how many individuals there are. So, the number of individuals in the population, honestly, can be really tough to measure. For instance, let's think about our snapping turtle. Do you think it's reasonable or feasible or possible to capture every snapping turtle in Pymatuning, it's a big lake, and count them and tag them? Probably not. That would be pretty hard to do. It's even worse for species that are really mobile. Think about wild horses out west. Think about insects. They're so numerous. How would you ever count them? Well, I'm glad you asked. We sample populations to get an estimate of population size when we can't count every individual. Once we've got a sample, the population can be estimated. We're actually going to do a sampling exercise, but first let's talk about a couple factors that can affect how a sample works. The first is population density, and that's how crowded the population is. So if we have 1 acre of land and it's got 15 rabbits on it, well, that's not a lot of rabbits, and that's a whole acre of land. What if we have 15 rabbits in a quarter of an acre? We have the same population size, but the land area is much smaller. So our rabbits are more crowded. It's a higher density population. What if we take those same rabbits and put them on 3 acres or 5 acres? Gosh, they've got lots of room to spread out. So we can have a low density population, a medium density population, or a high density population. If we think about humans for a second, cities are high density populations of humans. Rural areas like Columbiana County or Wyoming, where they have more cattle than people, are low density, and Wyoming is lower density than our population here. Okay, so could that affect how sampling works? Sure. You're going to get more individuals in the same sample net, so to speak, in a high-density population than you will in a low-density population. Doesn't mean the population size is different, but it does mean the density of the population is different. The other thing that affects how a population size looks when it's sampled is their dispersion, how they're spread out in their area. So not all species disperse themselves in the same way, and not all species disperse themselves in the same way all year or all day or all the time. So a clumped distribution is when we find flocks or herds or packs. We find organisms of the same species that are together a lot. We can think of packs of wild horses and packs of wolves. We can think of flocks of birds. Now, do all of these species clump all the time? Well, no, not necessarily. Birds clump for migration. You'll see great flocks of birds heading south. Packs of wolves may clump when they're hunting prey. They may spend other time wandering the landscape in smaller groups. Birds that are nesting may be in a group of 2. When they're rearing their young, they may be solo. It may be one parent sitting on the nest. So clumped distributions usually relate to either social behavior, I'd like to be near you, please, we're going to clump up, or sometimes it's because food or some other resource is clumped. If we think about African water holes, we'll find lots of zebras at a water hole at one time, so they're there for the resource, but they're also there for safety. That's clumped distributions. In an even distribution, we find animals spreading themselves out, well, evenly. This also usually relates to social interactions, but not the, hey, come here, I want to be near you part. Instead, it's the, hey, stay away from me part. We can actually find nesting colonies of birds where all the nests are spaced as far apart as possible, and they almost look like they were drawn with a computer. They're so perfect. It's a social interaction of the get away from me variety that causes an even distribution. Again, are these birds always arranged in that even distribution? No, but they are when they're nesting. Random distributions are a little less common in animals and very often result from seed dispersal by wind or birds. So we find this a lot in plant species. Plants where the wind carries the seeds or a bird drops their seeds. And we may not ever understand that there's no order to it. Random distributions can look like just about anything, but they make random sampling work really well to understand the size of a population. So again, how a population is dispersed will help a biologist understand how they should sample that population to get an accurate estimate of population size. And that's what we'll talk about more when we do our paper lab on sampling. Now, can you identify 3 things biologists measure about populations? Can you distinguish between dispersion patterns? Can you explain how density and dispersion might affect sampling? If you can't do this last one, don't worry, because we'll pick that up and cover it well together. Take care.